Good morning, good evening. Thank you for listening. This is a agricultural and horticultural gardening show focusing on plant science, soil science, and microbiology. And the information is for educational and scientific purposes. It's fact-checked. Any of the papers or websites that are discussed will be put in the description below. In today's last week of the Microbes and Agricultural course. It's going to be great. And I'm going to go through my latest microbial research that I'm doing at the end, which is quite up to date and interesting for those into plant science and microbiology. You'll like it. Excuse me. So if you're rewatching this back, fast forward until you see some slides or the screen change. I'm going to give it another 60 seconds for folk to arrive. Another 40 seconds. There's 10 seconds to go. Yeah, I'm starting. Righto, here we go. Microbial Bio-control Reproduction is the stuff on today's agenda. Actually, I'll just change the screen. The last four approaches, last four section. So traditional approach in biocontrol, pure culture microbes. This involves using single, well-characterized microbial strains that are isolated and cultivated in controlled environments. The strains are specifically selected for their ability for different pathogens and to promote plant growth. You're selected for different reasons. For example, Bacillus thuringiensis, widely used as a bioinsecticide, and it works really, really well for Trichoderma species. These fungi are employed by suppressing fungal diseases in plants, promoting healthy root systems and overall plant resilience. And if you're getting certain members, BT produces auxin, IAA, and Trichoderma variety does, and Trichoderma hazarium does not. So select your microbes well. Traditional approaches in biocontrol. Some advantages. Also controlled outcome, and you can get rid of specific problems relatively easily if you get the right biocontrol agent that can target it. It just takes care of it. You don't have to worry about it, and then it'll keep taking care of it if it's going to reproduce like spore form, like bacteria, most bacteria. But its limitations are pure cultures. There's a narrow scope of pure cultures that often do not have a limited range of activities and may not address complex pest and disease interactions. So for overall, actually, no, because there is some ones that do it overall, like Montgerensis is a biocontrolled pest to get different types of pests, insects. So it's a bioinsecticide. So that does take care of quite a few, but in general, they only do one or two things. So that's why you've got to have a few in your belt, a few at your arsenal to get different jobs done. Adaptation issues. Single strains may not perform well under diverse environments or in the presence of varying soil and plant microbiomes. So yeah, like last week we're going on about how microbe interactions, some are competitors and some are predators. Some like lactose, for instance, that puts out a heavy biofilm. That's why it's used in preservatives and food. So that one has a lot of competition against a lot of others. That's what that means. Shifting paradigms in biocontrol. Microbiome engineering approach. So this approach involves manipulating the entire microbial community associated with plants, including all the microbes as well. It focuses on enhancing or modifying the plant microbiome to improve plant health and resilience against pest and disease. Kind of what I do, because you could take any soil, any substrate, which is healthy, and improve it. That's what this is talking about. So if you want to get into that, there's the paper down the bottom, the DOI. Put those numbers in. That'll be a good read for you. That was written in 2020. What's the slide say? That's a comparison between the individual microorganisms-based reduction approach and microbial community-based holistic approach. It's hard to read. It's a shame. Oh. Still hard to read. Key strategies. 4 of them. Inoculation of soil in the rhizosphere. 2. Endophyte and seed microbiome engineering. 3. Host-mediated selection. And 4. Tissue and wound inoculation. So 4 ways to get your microbes into your plant and soil. And which ones are easy? Inoculation of soil rhizosphere is easy because you'll make most of the microbes are water soluble, meaning everything will dilute in the water. There'll be no residue left over. So that's where you'd pour it in, you'd make a ratio up and pour it in water and administer it that way. This one, endophyte seed microbiome. I should just read this instead of trying to explain what they're doing. I don't like doing that way. Modifying the microbiome with plant tissue or seeds to enhance resistance to specific pathogens. Endophyte seed microbiome engineering. So what that means is you'll get your right microbe, which is an endophyte, which lives inside of the cells, and that will transfer in the reproductive bits, and then for the seeds, the progeny that is produced should have those endophytes in there as well. That's why they say you can't import seeds. Host-mediated, so that's why also buying seeds or sourcing seeds or from an organic producer is way better for that reason. You're getting your microbes which work with that species already. Host-mediated selection. Host-mediated selection. Selecting microbiome communities based on the interactions with specific plants hosts to enhance plant defense mechanisms. Okay. Tissue and wound inoculation. Well, it says, pretty much says it. You'll scratch the surface off, expose a bit of the phloem or xylem, the bits that are wet, and inoculate that. And then that should get into the cells and be transported around through the plant. Yes, but the benefits is other things can get in too. That's the only problem about that bad one. So the best one really is this one, because develop that you'll slowly develop this anyway, and this way it's just easy. I should see if there's any questions. Hey, it's good. Genetic Memory Farm says, good morning, good peeps. I hope you're all safe and happy. G'day, mate. Hope you're safe and happy too. It's the last week of this course, so please suggest stuff to go on for next week. So I don't know what you're talking about. I got a Q&A at the moment. Where are I up to? No, you don't want to see how much solar I'm producing. Advantages of microbiome engineering: holistic management, enhanced resilience, and adaptability. So by doing all these things, by inoculating your soil and rhizosphere, you can get these type of approaches, these type of advantages. It's all around really good. And the challenges of doing it, it's very complex. What can be stability and persistence and regulatory and statutory and safety issues. So it's hard to know without your education. Well, now you've got this search engine. So I've just been explaining through these last few weeks how to get results by our search engines that we use. Taking photos or putting in the right words. And therefore you'll know what to source. Once you got that, source it. And that's the hard bit. It's really the challenges. And the safety issues, well, Bacillus cereus is good for food poisoning. What's another one? Bacillus ligniformis, that can do it. So you just got to be careful using them, really. Yeah. Future directions. The shift to microbiome engineering represents a forward-thinking approach to biocontrol, leveraging the natural interactions with microbial communities to achieve more sustainable and effective pest and disease management. Optimizing microbiome designs by developing methods to precisely engineer and monitor plant microbiomes for specific agricultural needs. Also, interact microbiome engineering with other practices by combining microbiome engineering with traditional IPM and understanding long-term impacts by studying long-term effects of your microbiome modifications on plant health, productivity, and ecosystem dynamics. That's pretty important. This is, of course, was written last year. Excuse me. So that sums up the future. So if you can really do a few of those things, you're up there with the leading scientists in what they're doing to try and move forward. Development of bacterial biocontrol agents. So how do you develop them? How can you reproduce them? This is a rad bit. Selection of antagonistic bacteria. So you can isolate it and screen it out, and it's to develop it. Okay, this is how they, for instance, you might want to get an azos out of your soil. So that's what you'll do. You isolate it and then screen it, identify it, then go through the blast system and it'll be identified. So this is the lab approach. And then once you've got the blast will come back and tell you your DNA and it'll match with this number of 97%, and then you'll be pretty right. And you've developed it, you know what it's like, then you just got to reproduce it. That's the bit I was sort of thought this was. And registering, okay, good. Development of bacterial biocontrol agents. Isolating and screening. Oh, that's kind of was. Effective sampling for diverse niches and proper selections of sampling media and isolation techniques are crucial. Bacterial antagonists are found in plant environments and bare soil. So that's why I say at the end I'll be going through my latest research, which is of the psychrotrophs, cold temperature environment. It's going to be pretty rad to hear that. I should have nearly talked about the start, but oh well. But it's the future. And if you can really do that, you're up there with what everybody's studying or researching at the moment. I only thought of it coming into this winter, and then I've looked into it in the research engine, and I'll talk about that more at the end when I'm describing what the psychotrophic bacteria. So developing strain selection, characterizing. That's where they'll upload the DNA data. Then the final stage is focus on large-scale production, formulation, and preservation. So this is where you go and buy your stuff off. So a retail outlet sources them from these places that do this process, which is what I was involved with for bacteria, beneficial bacteria. And here's an example, another screenshot for you, because it's got some genome sequences. So the numbers of specific, you'll look up a study and they'll have used FT333 of variety, Trichoderma variety. That's variety varans. Anyway, possible it's out. Azarium. So I think that's spot wrong, dude. But so examples of why this is important is because you'll read a study and it'll say that they used Trichoderma varieties FT333, and then you'll go, wow, how cool. I know what strain number they've used now. How do I get it? Oh, well, you get onto your mob and they go, oh, we can't get it. It's not accessible. You'll go, I've got the accession numbers. And then you'll give them that, and then they can source it from their wholesaler. So that's why these things are important. Published in and after 2015, so it's relatively up to date. Well, not to be honest, actually, not many of these studies have been done. Look, see, there's Bacillus cereus. That gives you food poisoning straight up. Look at all the different subtilis. It's one of the most studied around. It's even good for humans. Anyway, let's go on with it. References, that's it. So now it's up to my little bit. So I'll see if there's any questions. No? Righto, I'm going to get into it with my latest, what I've been doing. Excuse me. So it's at 17, 15. This is my latest research that I've been into. So here's a study on psychrophilic bacteria, Phosphate-Biofertilizer. So there's psychrophilic is temperatures, say, at freezing level where microbes interact. The next one up from that's mesophilic, which is about 20 degrees room temperature, or 65F, 70F. And then you go up to thermophilic, which is what you want in your compost heaps. So in winter, crops don't really function well because of nitrogen assimilation is slow. I'm not going to go into that. You get about 10% at 10 degrees Celsius, so 50F. So it still can happen. So to eliminate cold stress and to make this process happen faster, or like as if it's not cold for them, I got into this studies. So this is in the description below as well. So pretty much described what it is. So what happens is when the microbe, it's these different microbes, they will secrete enzymes that are, what's the word I'm looking for here? Cryoprotectant. That makes sense. Where I was up to actually, section 2. There? No, further on. The cold regions cover a massive amount of the Earth. So this is really good to get into. It does say some notable plant promoting bacteria inhibiting the extreme cold environment. Some genuses, Pseudomonas, Strachia. Anyways, quite a few of them. How do cold active bacteria survive under cold stress? Osmoprotection. That's what you're looking for. Because to thrive well under cold extremes and protect themselves from cellular injuries or to reduce the destructive effects of ice crystal formulation, microbes have developed various structures and functions. So if you can get these microbes, they will do your job for you and can turn crops that are not frost tolerant into frost tolerant. And all of a sudden you might get your different crops that are growing more vigorous from these microbes or these enzymes, as I'll get to a bit later. Because osmoprotection, antifreeze proteins. Oh, there it is, cryoprotective activity. At cold temperatures, the membrane of cold active bacteria becomes significantly more inflexible, which activates a membrane bound sensor. The sensor upregulates, and different genes are adapted. It upregulates different genes that assist bacteria to adapt to the cold. So, therefore, you think, "Ooh, how cool is that?" Well, maybe I don't need these microbes. What about I just get types of cold active plant enzymes? What types are they? Maybe I can source them. Or maybe, because those types or key types of cold active plant enzymes include pectinase, protease, amylase, and lipases, which is unique roles in breaking down specific substrates as well. But they're cold active plant enzymes. You want active, not passive. Ones that be active and they can actually do jobs for you. And then you'll think, well, is there anything that I could read on that? Well, there is. Here's a study that was released a few months ago on cold active microbial enzymes and their biotechnological applications. So that's what I'm into at the moment. And you can see it's quite, I can't see that anymore. All right, I'll bring up the I don't want to download it. I have to. All right. Righto, what? Nothing happened. So why they won't show me the to show me. It's a bit bigger. Cold active enzymes. Here's some study. Remember I was talking before about that sheet, that why would you want that sheet? Because after reading some, for instance, bioremediation, leather and textiles. It's hard for me to give an example. Well, if you're reading these studies, you can go through and get the actual one that you want if they're listed in these studies. Here's some important cold enzymes and their microbial source. So I was saying last show that I really would like to go to a snow or glacial environment and just scoop up some of their substrate and rear that. So make a tea out of it and try and reproduce some of those microbes that are in there. Because those are the ones that will, where's an example? Cryobacterium. Wow, you know what that is? That's cool. Subglacial under 5 Celsius. That's rad. I was looking for a name that might be familiar that I could relate to. Trichoderma gamesy. The trichoderma wonder. Well, there actually it's perfect because in 2023, Wang did this study on biochemical properties of cold active chitinases from marine Trichoderma gamesy. This one. And the chitinase produces the enzyme which works in the cold weather. So for you to source this exact microbe, you want the R1 and you will get this outcome. That's a way better example of how I was trying to get to before. Remember I said I'll wait till later on? Psychrobacter. That's rad because a psychotroph is a microbe that works around the freezing levels, and Psychrobacter They just thought they'd name that one, and it works in 0. Look at that. Actually, freezing level, 37 Fahrenheit. Found in Siberia. Now, interesting. This is so fascinating, because what happens if we can get a fair bit of these exact ones, because the ones you'll find that, yeah, that's a good time to talk about that too. Trichoderma gamesii, I can probably get that in Australia, and it won't be the R1, so it won't be putting out chitinase and doing the job that it should be as a cryoprotectant, which is what I would like it to do. So by sourcing them in these cold environments, they're pretty much developed the strains over years and decades and years and years. I mean, microbes are billions of years old with a B. So from sitting in those environments over billions of years, epigenetics has just slowly developed their genes to adapt. This is a decent list here. I'll be downloading this list. Bacillus pumilus. See, there's an example. I use that one. And people would think, oh, rad, I use that one. It's going to be putting out, well, actually, it might put out protease, but it mightn't be a cryoprotectant in the sense that I'm looking for. This is cold active enzyme in food. Okay, now it's going to break it down a little bit more. This is cool. Yeah, I haven't read through this one. Another one for me to read. Oh, I'm not sharing. You're joking. I'm not sure. Did I not share that whole thing? The last 10 minutes I've been talking about this example. Oh, no, that's right. I did stop to ask questions, and it was the last little bit that I wasn't sharing for, so that you won't see. You've seen pretty much all of it, except for, well, I assume, except for this list. So if you wanted this list, you can get this paper in the description anyway, and you can go and download the list afterwards. But this is the list I was talking about, how different microbes in these environments have specific strains, and that those strains are relevant to the cold weather, where the strains that I can get of exactly the same species of microbe, it's more than likely will do another thing. And on the left is all the enzymes that it puts out. And I like this one because it actually mentions the temperature. 0 is freezing, so 37 F. 5 degrees F is 40 Fahrenheit. So if you're living in North America, Canada, States, go to your glacial areas, get some of the soil, put in a tea, and spread it in your garden. And you might get some cryoprotection and some benefits in the wintertime. If you get a sudden frost, it might be frost tolerant. And then you'll go and develop seeds from that, which will change your genes. So you bioengineered a different species. I suppose it could be species because the DNA tests different. But you've gone and altered it to your advantage. That's what this is all about. It's pretty cool. So I'm still sharing. Good. Then it goes down. Okay, to talk about each enzyme in the cold environment, how it works. Like amylase, it breaks down starch. It's one of the ones in the seeds. Remember that the seed puts out, because most of the seeds starch, so it can use that as energy. Cold active amylase. And Antarctic bacterium was the first microbe producing cold adapted alpha amylase, which was extensively studied and been magnificently expressed in a mesophilic host, E. coli. Wow. This is cool. This will all be happening. These will be available probably next decade. In the 2030s, you'll see all of these microbes will be available and people will be growing in cold weather. It's that easy to do. Mind you, it's easier said than done because I've got no access in Australia where I can go and do it either, unfortunately. We don't have glaciers here. We have cold environments, but there's no real glaciers that would harbour these types of microbes. So I'd have to be looking to import the strain names of this list that was up here, of some of these ones, if I was really interested. And then for me to do that, I'd have to go, well, okay, why do I want protease? What's it breaking down actually? So I want more of a cryoprotectant, and that might be like the stuff you put in your radiator in your car, the glucan and that stuff. This is throwing me off track now. Nitrous oxide reductase. Like I don't know how that is worked as a cryoprotectant. Or is it osmo? Is that word again that they osmoprotectant? That the word? Yeah, osmoprotection. So that's how I got to that, how I found out for this. So you people in the cold environments can get an upper hand in the studies that I'm doing. Look at this one, 22 Celsius. Why is this in here? See, that'd be good to read that. Production, purification, and characterization of cold active lipase from a Psychrophilic Pseudomonas species. But yet the temperature of habitat is 20 Celsius. That's pretty interesting. I wonder what difference. Look how big they've got it now. I used to just do this and just see if you can read it, and it's nearly the whole page. You've got no blimmin' choice. This is what I want, figures and references. I like them. There they are. So that's one tested. Pseudomonas petita. Do I use that one? No. It's familiar to me now. Azoteforms. Wow, that's azo suggests that it might produce nitrogen. I don't know any of those. Unfortunately. Production purification practices. Other references is there. I got them. Deep frying. Kind of studying out loud here, doing research. This is how I'd look through blotting chart. Okay, it's just after the names of them, and see the strain numbers after them. Very, very important for these types of studies. So Tita MT. So from my source, I would try and look up the MT 626824, and I might have some luck in cold environments if it's the same condition. So see how they've grown it here? They've inoculated it and they've reproduced it, and the temperature, 10 degrees, has led to maximum lipase activity. And they got 23 microliters per milliliter. That's rad. 10 degrees Celsius. That's the best bit. It's a psychrophilic that you can grow, reproduce, and it's even gives you the formula. So, oh, wow, soybean, 7.5% soybean oil as the base media. That's a fair bit of weight, isn't it? Huh. Soybean oil. Jeez. It's a new one. See, all these science, it just keeps evolving. So I love it. Soybean. Cool as. Here's some other similar. Anyway, I should see if there's any questions. Any questions? No questions. That's unfortunate. Hope you're enjoying it. If you're enjoying it, please press the like button. And if there's any suggestions for shows for next week, please add them, because I don't know what I'm going to be talking about. Might have a Q&A. That's what I've got it down for. So if I get no suggestions, I'll just have a Q&A. Oh, discover low sodium use. I use Pseudomonas fluorescens. Still subtilis is always in everything. It's probably even on the scheduled list where you don't even need an import thing now. DNA extraction, culturing of lipase, seed cultures. Lipase activity. Very good. Statistical experimental design, not bad, it's not results. Purification. Results. 7 bacterial isolates capable of growing at 10 Celsius or 50 F were isolated directly from seawater. Only a strain named A6 displayed growth at 10 C. After all, recovered isolates were examined for their best growing temperature. The ability of A6 to produce lipase was assessed qualitatively. As a result, the isolate was chosen for future studies. So they found one, at least they found one, A6 out of 7. A6 Pseudomonas. You got it. Oh wow! See down here, it shows the Pseudomonas A6, and then it shows all of them in A6, which is all these. Wowee. Blast revealed that it had 100% similarity to the genus Pseudomonas. There you go. It's great, this stuff. I love these things. You can solve your own solutions and do results and get results regardless. So the carbon source, that's why they use soybean oil. So fructose and deep frying waste is in here. They've even used that as a growth media. Deep frying waste. Of course, you put it through your autoclave and sterilize it, but man, that's just amazing. All these, coconut was one of the first, that's how they, with tissue culture. Look what they've done now. That's great. True science. If you don't stay up to date, I'm only out of tissue culture for probably 12, 18 months at the most. That's cool as productive purification. Okay. Because once they, in the lab, by culturing up these secondary metabolites, they get nano amounts. And then they've got to go and, like, if you buy a essential oils bottle, it'll come out with very concentrated. So you've got to purify it and, what do they call that? What do you fold it over and over? Anyway, that's what they got to do. So that's what. So you don't get much out of these actual productions of secondary metabolites, but it's the overall summation of them all, and you do it over and over, and it keeps working. cold-adapted proteins and enzymes are found in psychrophilic bacteria, which allow them to keep metabolic activity at low temperatures. That's the whole. That's what I would like to have said at the start. Actually, copy that. I can go. I'll put that in later on. That's exactly why I'm interested in it. Proteins and enzymes. I didn't mention proteins. I'll leave that highlighted because I like that. Recently, many publications have been reported. Pseudomonas fluorescens, yeah, KE38. Let's see what one I use. And then they found that A6 grew. That's right. It's just about alpine. I like the snow. Similar findings were seen for lipase production in neutral to alkaline media conditions by psychrophilic bacteria that were isolated from alpine regions. So yeah, there's something to do with different micro strains up there. Let's go. And there you go. I proved it. Not proved it. I've shared it with you so you can do your research as well. About proving nothing. Science is just science, forever changing. Okay, well, I'll put those papers in the description. I'll give it a bit, add some questions if you got any, please, or discussion topics, because the show's going to end in a few minutes. I'll give you a chance to come through, think of anything. There's a couple of people watching. And press the like button if you enjoyed it. I love talking about microbes because they're the future. Make microbes work for us. So cool. Yeah, that'll do. I'll skip just talking about microbes. So next week is going to be a question and answer. So I'm going to sit on the screen and probably just, I don't know, but I've got no suggestions to do any topics. So I'm going to do that. So hopefully there'll be a few Q&As there. And if it's a short show, it's a short show. I haven't had a short show for ages. I've had all these topics, these awesome topics. And I've enjoyed the slides. Very cool. GMF says, if I can think of any for next week, I will holler. Yes, thanks, mate. I messaged you, gave you a message too on Zoom. And I'm not sure really, but I don't mind the Q&A for a bit. That'll give me a bit of time to think about stuff too, because my studies start up in 4 weeks' time. And I don't know about you, but you folk in North America, I hope you're getting into the cricket. The MLC's on, Major League Cricket, and it's cool as. There's heaps of international players playing, and 6 states have got a team now, and there's going to be way more. It's going to get big too. I might even take over MLB. Oh, look out, baseball fans. People are swearing at me at the moment. No. It's a international sport. It's very popular. GMF says, popping seeds soon, so it will likely be that. Oh, very good. So yes, some inoculation questions. That'll be all right. I can maybe... Actually, yeah, that's a good idea. Ask that, GMF, and I can... I'll go through some nanotechnology about seeds, what you can inoculate them with. Do your own of A, B, C, 3 different strains of your different seeds. One can be with pyrite, one can be with something else, and zinc. I don't know, something else. But yeah, I admit, never watched cricket. Would you watch cricket? Just says GMF. Never watched it. They were, one bit I like is I watch it every day. There's a match on right now. When we finish here, I'll be watching the match, Texas Rangers versus I think it's LA Knight Riders. Goes for 3 hours. It's an hour into it already. No, it's 2 hours into it. So there's an hour to go. But for today's match, and then tomorrow there's 2. So that's rad. But anyway, yeah, they're interviewing the crowd, and the crowd people are just saying differences between baseball and cricket. And yeah, it's cricket's more of a joint sport. Like the teams are very friendly with each other. They shake hands, they talk to each other on the field, and they laugh and carry on. Where they're saying with baseball and other sports at these coliseums, it's very one-sided and not as fun. Well, cricket's more funner. And I was very happy to hear that, because I really like cricket. I watch it I watch a few different leagues around the world. Watch with you, sure. Yeah, I could watch it. Message on Zoom, and when I watch it, I'll help you with the instructions, because it's funny, and they interview the crowd a lot, which is what I like, and you get some funny remarks from the crowd, and these little kids I'm going to get into cricket, and their favourite players, and it's really enjoyable. It's a good all-round sport. So I just wanted to share that, because I know most of the people that watch this stream are in the North America. And after the North American League, the USA League, goes the West Indies League in Barbados, down below. So that's cool. And I can't do a live stream on YouTube with the cricket, because I'll get a ban thing. I'm not allowed to do that, because it's a paid telecast type thing. And yeah, I got in trouble ages ago from doing the surfing, which I got a big strike on that. I said 3 of them and your channel's gone. And I've already had 1, so I don't want to risk doing another one. Yep. Yeah, that's the go. So next week will be a question and answer, possibly a seed thing, possibly. See how he feels. See what other people in chat reckon as well. Like to help out as many people as possible. And thanks today. I think that's it for today. I'll just be otherwise talking about cricket. We got cricket on right now. Woo! Let's go watch it. I'll check it out and tell you actually who is playing. I'm not sharing. Make sure I'm not, because I'll get in trouble sharing. There it is. Yeah, it's Texas Super Kings and the San Francisco Unicorns. The match finishes in just over an hour. I'd love to share the screen and blim and play it, but I can't. If you don't get the, you don't need to pay for it either, and that's how I don't get it. I don't pay for it, but there's a lot of ways to get it without paying for it. Actually, I better not go down that route. Best thing is, if you really want to watch it, put in the Texas Super Kings versus the Willow by. Oh, where's this? I'm not sharing, no. There we go. That's the that's what's on right now. So if you really were interested, you can put that into your search engine, and that might come up with suggestions on how to watch it online for free. But yeah, that's it for today. Thanks everyone for watching. I hope to see you here next week. See you, brother. Be safe, says GMF. Thanks for your nice words, mate. And everyone, I hope happy breeding, happy growing, and good health to you all. You have a fantastic week and weekend. Bye-bye.